Hello and welcome back. This lecture will start the first part of Chapter 7, aptly titled The Cell Cycle and Cell Division. Here in this lecture, we will learn about the life cycle of the cell and the process of how a cell divides into cells. So let's get started. The lifespan of an organism is linked to cell division. How so, you may ask? Cell division is important in the growth and repair of a multicellular organism and reproduction of life. In cell division, a parent cell divides its genetic material and then divides into smaller cells. The picture above is a cell undergoing cell division. Yellow lines are highlighted spindle fibers that help facilitate this, as we will see later. As mentioned earlier, cell division is important in the reproduction of single-cell and multicellular organisms. It's important in the growth of organisms, as shown in the root tip in the top right, and in the regeneration or repair of tissues, as for our poor lizard friend on the bottom who lost its tail. Organisms have two strategies for reproduction. They are asexual reproduction and sexual reproduction. In asexual reproduction, the offspring are clones, genetically identical to the parent. Any genetic variation is due to mutations from environmental factors or copying errors while DNA is being made. Single-cell prokaryotes usually reproduce by binary fission. Single-cell eukaryotes can reproduce by mitosis and cytokinesis, as we will learn about later. Many multicellular eukaryotes can also reproduce by asexual means. Here's a picture of aspen trees that are genetically identical to one another. This is an example of a multicellular organism reproducing by asexual means. Next, we're going to talk about sexual reproduction. Sexual reproduction involves the fusion of gametes. This results in offspring having genetic variation, unlike asexual reproduction. Gametes form by meiosis, a process of cell division that reduces genetic material by half. And we'll talk more about meiosis in the next lecture. Within eukaryotic cells, DNA is organized into chromosomes. Body cells not specialized for reproduction are cells that aren't sex cells. Examples of these would be nerve cells, epithelial cells, muscle cells. These are called somatic cells. Each somatic cell contains two sets of chromosomes that occur in homologous pairs. One homolog comes from the female parent, while the other comes from the male parent. Homologs have corresponding genetic information. You can see the chromosomes above. Humans have 23 pairs of chromosomes. Pair 15, for instance, has one chromosome from the mother and the others from the father. On these chromosomes are the genes for eye colors, as well as other traits. Gametes only have one set of chromosomes, meaning they are not in pairs as somatic cells. They are referred to as haploid cells, or denoted as N. When fertilization of the two haploid gametes fuse together, they form a zygote. Zygotes are diploid, meaning the chromosomes are paired with their homolog, and denoted as 2N. An example of a diploid organism are you and me. All sexual reproduction involves meiosis. In some organisms, gametes such as sperm and eggs develop immediately after meiosis. Fertilization results in a zygote, which starts the diploid stage of the life cycle. Some organisms, such as ferns, have a diploid and haploid life cycle, but many animals, including mammals, have a diploid life cycle that dominates their entire life. The diploid life cycle occurs after the haploid gametes fertilize. One of the benefits of sexual reproduction is that the chromosomes that end in a gamete are random, and each gamete that is produced only has half the parent cell's DNA. For example, look at the picture at the top. The red chromosomes are from the mother, and the blue are from the father. During gamete formation, as we will see later, the gametes have half of the parent cell's DNA. The gamete will most likely have some combination of the mother and father's DNA in them. This helps make the gamete cells have variety. When two gametes fuse together, they make a diploid cell that is genetically different than the parent cell. This is important in shuffling genetic information in populations, where no two individuals have the same genetic makeup. Here's our first progress check before we get into the cell cycle. Please take your time to review your notes and answer these questions. Move on with the lecture whenever you're ready. With that background information out of the way, let's jump into the cell cycle. With any type of cell division, there are 4 events that need to happen. First, there needs to be a reproductive signal to initiate the cell division. Next, DNA needs to be replicated so that the daughter cells have the genetic information. Third, the DNA needs to be segregated and distributed evenly to the 2 new cells. And lastly, cytokinesis needs to occur. Cytokinesis is the division of cytoplasm and the separation into 2 new cells. Before we learn about cell division in eukaryotes, we will focus on prokaryotes. As mentioned before, prokaryotes divide by binary fission. The reproductive signal that occurs may be an environmental signal, such as factors that favor bacterial growth or nutrient availability. Most prokaryotes have a single circular chromosome with 2 important regions for division. There's the Ori region, where DNA replication starts, and the Tera region, where replication ends. Replication occurs as DNA is threaded through a protein structure called the replication complex. As DNA replication proceeds, the Ori complexes move to opposite ends of the cell. Figure 7.4 diagrams binary fission in a prokaryotic cell. As the DNA is replicated, you can see the replication complexes, which are the red dots illustrated here. They will move to opposite ends of the cell, thus pulling the replicated DNA with them. And at the end of binary fission, you have 2 new bacteria cells. Next is cytokinesis. After the DNA is replicated and the DNA is segregated, the prokaryotic cell membrane will pinch in on itself by using a contraction ring of protein fibers. As the ring forms, a new cell wall will be deposited, resulting in the separation of 2 cells, thus completing binary fission. Eukaryotic cells divide by mitosis, then followed by cytokinesis. Reproductive signals usually tell the cell when to divide in eukaryotic cells. Before mitosis, replication of each chromosome must occur. During mitosis, the replicated chromosomes need to segregate, with the end result of the two cells forming from one cell. The cell cycle is the period from one cell division to the next, and it's divided into two main stages: the M phase for mitosis and interphase. The mitosis phase is the segregation of chromosomes into two nuclei, followed by cytokinesis. Interphase is where the cell nucleus is visible and cell functions occur, including DNA replication. Figure 7.5 shows a diagram of the cell cycle. You can see that interphase is a majority of the cell cycle, with mitosis not taking much time at all. However, this isn't to downplay mitosis, as we will see it is very important in the formation of new cells. Within interphase, there are three subphases: G1, S, and G2 phases. G1, which stands for the gap 1 phase, is variable among cells. It may last a long time in cells such as ones that aren't actively dividing. During this phase, the cell is going about its day-to-day business, growing, synthesizing proteins, and just being a cell. The S phase, which stands for synthesis, is where DNA is replicated. G2, or gap 2 phase, is where the cell prepares for mitosis, such as synthesizing microtubules for segregating chromosomes. During interphase, DNA is packaged tightly around proteins. Kind of looks like a bunch of spaghetti noodles, usually in diagrams. During mitosis, the chromosomes become much more tightly coiled and condensed. During this time, you can see the chromosomes underneath the microscope with the aid of some stains. After the replication of DNA during the S phase, each chromosome has two DNA molecules called sister chromatids. Chromatids are what DNA is called when it's condensed tightly together. Sister chromatids are identical to one another. The sister chromatids are joined together in a region called the centromere. You can see in an electron microscope picture a DNA molecule. The left side is one DNA molecule, while the right side is another DNA molecule. They are both identical. These are sister chromatids with a centromere in the middle. The karyotype is the number and visual appearance of the chromosomes in the cell nuclei. Pictured here is a karyotype of a human. The chromosomes that you see are condensed DNA. The centrosome is a region in the cell that consists of centrioles, which determines the orientation of the spindle fibers and will aid in the separation of the DNA. The centrosome is duplicated during the S phase along with the DNA. During the transition between the G2 phase and mitosis, it will move towards opposite sides of the nucleus. The centrosome position determines the plane of cell division. The centrosomes serve as poles towards which the chromosomes move to. The spindle forms between the poles from the centrosomes. During mitosis, the sister chromatids attach to microtubes from opposite sides, so that the sister chromatids become separated into individual chromatids. After they are separated, the sister chromatids are referred to as daughter chromatids. Okay, with all that terminology out of the way, it's important to know the difference between centrosomes, centromeres, chromatids, sister chromatids, and daughter chromatids. So please understand what each of those terms mean. It will help you understand the processes of mitosis. So with all that out of the way, let's talk about mitosis. The stages of mitosis in order are prophase, prometaphase, metaphase, anaphase, and telophase. I use the acronym PPMAT to help remember them for prophase, prometaphase, metaphase, anaphase, and telophase. You are expected to know what occurs in each of these stages in the order they happen in. In the first stage of mitosis, prophase, three structures appear. Chromosomes are condensed within this stage. The centrosomes are reoriented to opposite sides of the cell, and spindle fibers appear that will help to move the chromatids. In prometaphase, the nuclear envelope breaks down, and the chromatids attach to microtubules. In metaphase, the chromosomes, which are still sister chromatids, line up in the middle of the cell. In anaphase, the chromatids are separated, becoming daughter chromatids, and move towards the poles of each of the cells. Figure 7.6 is a diagram of the stages interphase through prometaphase. You can see interphase on the left, where the DNA is not condensed, and the nuclear envelope is pretty much still intact. In prophase, which is shown in the middle diagram, chromosomes start condensing into chromatids. Here they are sister chromatids. In prometaphase on the right, those sister chromatids start attaching to microtubules, as well as the nuclear envelope breaking down. Metaphase is pictured here on the left. As you can see, the sister chromatids are lined up along the middle. Anaphase is shown in the middle picture. The sister chromatids, which are identical to one another, are split into daughter chromatids with the help of the microtubules. The daughter chromatids move to opposite sides of the cell. Telophase is the last stage of mitosis. In this stage, the nuclear envelope forms around each set of chromosomes. Two nucleuses appear and the spindle breaks down, with the chromosomes becoming less compact and less dense. Telophase is followed by cytokinesis, which is the distribution of cytoplasm between the new cells. In animal cells, the cell membrane pinches between the nuclei. A contractile ring of actin and myosin microfilaments form, with the proteins contracting to pinch the cell in two. Top left diagram shows two animal cells undergoing cytokinesis. Plant cells undergo cytokinesis, but is a little bit different, as we will see in the next slide. In plant cells, vessels from the Golgi apparatus appear along the plane of cell division and fuse together to form a new cell membrane. Content within these vessels also contributes to the forming of the cell plate, which will then form the cell wall for the plant. Unlike animal cells, we do not have cell wall, so we do not have that step. After cytokinesis, each daughter cell contains all of the components of a complete cell. Chromosomes are precisely distributed so that each cell is genetically identical. Table 7.1 illustrates the major events that occur in each stage of the cell cycle. This table is good for your notes and a good review of what happens within the cell cycle, especially mitosis. Here's our progress check to end the chapter. Take your best shot at answering his questions. I do have a video of mitosis posted on Canvas. Please watch it after this lecture. It illustrates all the steps that we went over. By seeing the chromatids move and cells split, help understand what occurs within mitosis. Let me know if you have any questions, and that ends our lecture for today. Thank you for listening, and have a good rest of your day.